Hello, my name is Joshua.
My name is Justin.
And today we are talking about the enzyme PETase.
We're going to cover some backgrounds of PETase, how PETase was discovered, and a PET-consuming bacterium called Ideonella sakaiensis. We are also learning about the mechanism and future applications of the plastic-degrading enzyme.
Polyethylene terephthalate, PET, is a type of plastic widely used in modern society, but large amounts of PET waste cause severe environmental problems. The enzyme PETase exhibits superb hydrolytic activity and substrate preference towards PET. PET hydrolyzing enzymes decompose PET into its building blocks, providing an eco-friendly solution to PET accumulation in the environment. PET is one of the most extensively applied synthetic polymers and is composed of ester bond-length terephthalic acid (TPA) and ethylene glycol (EG). It is a popular material used in the production of drinking bottles and fabric because of its transparency, malleability, and resistance to the natural degradation processes. The annual production of PET is estimated to exceed 50 million tons. Large amounts of this plastic accumulate as waste in landfills and oceans and have caused a severe environmental burden. Using enzymes to decompose PET into its building blocks is an eco-friendly strategy to reduce plastic waste and recover petroleum-based starting materials TPA and EG, ultimately closing up the loop of PET production and recycling.
The first PETase enzyme was discovered in 2016 from bacterium Ideonella sakaiensis, found in sludge samples collected close to a Japanese PET bottle recycling site. In the last decade, a number of enzymes from plastic-degrading microbes which can cleave ester bond linkages, including lipases, esterases, carboxyl esterases, and cutinases, have been reported to hydrolyze PET. Among them, cutinases are the most promising PET hydrolyzing biocatalysts due to their higher efficiency and lack of the lid structure that lipases require for activation at lipid-water interfaces. Cutinases belong to the alpha-beta serine hydrolase family and employ a canonical catalytic triad consisting of serine, histidine, and aspartic acid to carry out ester bond hydrolysis. PET and cutin share minimal structural features other than the ester bond, which is attacked and cleaved by the enzyme. The real substrate binding mode of PET hydrolyzing enzymes still remains unexplored, but here's a video to give you a better picture of PETase.
So we need to find more effective ways to recycle plastic.
So this enzyme was first used in the UK to degrade PET.
This is a scanning electron microscope of the enzyme digesting plastic.
And it digests in about 96 hours into its building blocks.
How does the enzyme work? The enzyme is purified and crystallized.
The best X-ray crystals are used and applied to PET bottles, which then degrade.
The best crystals are transported to the diamond light source.
And here's the structure of it as well.
PETase is presumably evolved from cutinase, an enzyme that breaks down cutin, a waxy substance on the plant surfaces. This is miraculous, as PET is new, and so the evolution must have occurred incredibly fast. In an attempt to determine the structure of PETase by modifying PETase's active site to look more like cutinases. Scientists accidentally improved the efficiency of PETase by 20%. This reveals that PETase has potential for more engineering and accelerated reaction rates.
The bacterium Ideonella sakaiensis 201F6 can utilize PET as an energy and carbon source. Sakaiensis secretes a unique cutinase-like enzyme, PETase, to hydrolyze PET to BHET, MHET, and TPA, the building blocks of TPA. Under physiological conditions, 30 degrees Celsius and a neutral pH, the enzyme is 5.5 to 120-fold more efficient than previously reported PET hydrolyzing homologs. PETase exhibits preference for PET over p-nitrophenol-linked aliphatic esters, the surrogate compounds for lipase and cutinase activity determination. The fact that PETase and other PET hydrolyzing cutinases share high sequence identity indicates the existence of critical structural features responsible for substrate binding. Since even small differences between these enzymes are crucial, high-resolution crystal structures are needed to gain insight into the PETase substrate binding mode.
Here is the reaction that occurs on the PET plastic surface. PETase secretes, degrading PET into MHET, TPA, and BHET. In the journal, the authors summarized the most recent studies of crystal structures of PETase and its complex with substrate product analogs reported by them or other groups. Several critical structural features of PETase proven to be related to the promising PET hydrolytic activity are described. A mechanism of action of how PETase binds to and hydrolyzes PET is proposed. These results further research of the molecular mechanism of PETase, enzyme engineering, and identifying more potent PETase-like biocatalysts. We are going to explore the unique structural features of PETase.
Several unique structural features revealed by inspecting the apoform PETase structure could provide insights in understanding the mechanisms of PETase. First, PETase harbors two disulfide bridges, termed DS, whereas the other homologous enzymes have only one. DS2, which links the C-terminal loop and the last helix, is strictly conserved in all homologous structures. The PETase-specific DS1, on the other hand, is located adjacent to the active site and connects the beta-7 to alpha-5 and beta-8 to alpha-6 loops that harbor the catalytic acid D177 and catalytic base H208. In addition, the beta-8 to alpha-6 loop in PETase is 3 residues longer than that of other homologous enzymes. This extended loop is proposed to yield a subsite to bind PET, while shorter loops found in homologous enzymes may hinder the subsite formation. Interestingly, PET hydrolysis measured under reducing conditions in mutagenesis experiments indicate that DS1 depletion significantly reduces enzyme activity, suggesting that DS1 plays a critical role in PETase activity. Molecular dynamics simulation suggests that DS1 depletion may influence the distances between the catalytic triad residues and thus compromise the active site integrity. Scientists recently solved the crystal structure of the C210A variant PETase and found that the regions 176 to 178 and 205 to 216, along with the catalytic acid and base D177 and H208, were not visible, and that the substrate binding pocket was disrupted. Taking this data together, it is probable that PETase carries a longer beta-8 to alpha-6 loop to yield a larger space to bind PET, and that DS1 is required to keep the extended loop and the catalytic triad residues in their functional locations.
The discovery of PETase is a big boost on the route to designing a biorecycling scheme for synthetic polymers. To get insights into the molecular mechanism of PETase, solving high-resolution crystal structure of the enzyme is of vital importance. Residues which do not directly contact the substrate could play a role in enzyme catalysts. These results could only be obtained through in-depth crystallographic analysis. Other ester bond-linked synthetic polymers which are utilized as bio-based PET replacements also may be a substrate of PETase. PET crystallinity should be taken into consideration as a higher degree of polymerization may lead to higher resistance to the enzyme hydrolysis. PETase has been engineered to enhance activity by reducing the crystallinity. Another promising direction would be to engineer the enzyme so that it can operate at higher temperature where the degree of PET crystallinity is lower. Despite many issues left to be addressed, many more applications involving PETase are foreseeable. A deeper understanding of the molecular mechanism of PETase will provide an ideal starting point for the future identification of related enzymes.
And finally, here are references, and thank you for joining us and learning more about the enzyme PETase and its applications.